Here in the genetics lab, we obviously work with DNA. My project is trying to analyze an ancient Roman population excavated from the city of Palencia on the island of Majorca, Spain. Dr. AR, myself, and 3 other students traveled to Palencia to participate in the archaeological excavation ourselves. We were working in the field for 5 to 6 hours a day and then washing the bones and pottery that we found for another couple of hours.
Typically, from Christian background, we're buried on our back with our hands crossed. They also came across skeletons that were born in more of the Islamic style, where you're on your right side facing Mecca.
So we're trying to look at this population and use DNA to answer the question, where did these people come from? We start off with the human bone, and we don't have them yet. They're in the process of being exported. So this is just a sample. But once we do get the bones, what we'll do first is a very thorough decontamination procedure, and we'll drill into the diaphysis or shaft of a long bone to obtain some bone dust for the sample. Once we have that little bone powder, we will perform DNA extraction and isolation on it using a kit made specifically for ancient DNA. So that'll give us the DNA in solution by itself. Once we do that, then we need to amplify the DNA that we want to look at. And at that point, then we can send the sample off for sequencing to an external company, and then they will send us back sequencing chromatogram data, which we can read and actually get the sequence of DNA itself. Then we can assign kind of ancestral origin to these individuals.
Some of the work we do will help contribute to databases worldwide, so other researchers will be able to look at it and answer some of their own questions.